Pseudomonas aeruginosa. In this session, we are going to discuss Pseudomonas aeruginosa. You will hear a lot about this bacteria while working in a hospital. It is very important in hospital environment. Very important. Very important for two reasons. Number one, it infects and colonizes immunocompromised, sick, hospitalized patients. And these are people you are going to take care of them. And Pseudomonas aeruginosa infects them. Number two, this bacteria, this rascal bacteria is resistant to almost every antibiotic. Okay, but don't think that we don't have tools against this nasty bacteria. We have successful treatment options that we will discuss later on in this illustration video. Okay, so Pseudomonas has very important role in nosocomial infections. Nosocomial infection means hospital associated infections. It causes very severe life-threatening infections. Very severe life-threatening infections in immunocompromised individuals. It can also cause infections in healthy individuals, and it also causes infections, chronic infections in cystic fibrosis patients. With this information and with these facts, you are able to appreciate that Pseudomonas aeruginosa is a very successful hospital pathogen. What ecological factors make Pseudomonas so successful? Okay, so to appreciate the factors which make Pseudomonas so successful, we discuss its medical ecology. Pseudomonas aeruginosa is primarily a nosocomial pathogen. It is ubiquitous water and soil organism. It is commonly found in most environments like respirators, humidifiers, sinks, water baths, showers, and other wet areas. Flowers and raw vegetables are also contaminated with Pseudomonas aeruginosa. To know the habitat of this lethal nosocomial pathogen is important, it's critical. It is important in its prevention. Like, you must not allow flowers and vegetables in burn unit. Why? Because there are immunocompromised sick burn patients, and these sick patients are potential candidates of Pseudomonas infections, and raw vegetable and flower have a lot of Pseudomonas aeruginosa on them. That's why you must not allow flowers and vegetables in burn unit. Pseudomonas aeruginosa is found almost everywhere in the hospital. It is even found in the disinfectant. Survive happily and proliferate avidly in water with minimal nutrients. That's why it's found everywhere, almost everywhere in the hospital. So any non-sterile fluid in a hospital may be contaminated, may be heavily contaminated with Pseudomonas aeruginosa. It might be there. It might be in the humidifier, in the respirator, it might be in the contact lens, it might be in a medication, it might be in the disinfectant, it might be there in antiseptic, detergent solution. Sinks and aerators are heavily contaminated with Pseudomonas and serve as a source of infection. I am not scaring you. Actually, these information would help you to understand the magnitude of problem and would help you to appreciate the importance of preventive measurements. Now let's focus on to the bacteriology of this pathogen. So we describe the general classification and put the Pseudomonas aeruginosa in it. You know we broadly divide bacteria into gram-positive bacteria and into gram-negative bacteria, and we further divide gram-negative bacteria into gram-negative cocci, gram-negative rod, and acid-fast bacteria. And we divide the gram-negative rod into three categories based on the oxygen requirement. There are facultative gram-negative rods, and there are anaerobe gram-negative rod, and there are aerobe gram-negative rod. And Pseudomonas aeruginosa is aerobic gram-negative rod. After this brief overview, let's have an in-depth view of the bacteriology of this bacteria. Like other enterobacteria, it is gram-negative. It is gram-negative rod. But unlike other enterobacteria, it is obligate aerobe. It means that it derives its energy from a sugar by a process called oxidation. While other enterobacteria derive the energy from sugar by a process called fermentation, not oxidation, fermentation. So make it clear in your mind that Pseudomonas is non-fermenter. Pseudomonas has a lot of cytochrome C oxidase. Cytochrome oxidase C. This is an enzyme due to which it do the oxidation of sugar. This is the reason why the oxidase test is positive with Pseudomonas and negative with other enterobacteria, with other gram-negative rod. Again, Pseudomonas aeruginosa is positive in oxidase test, and this test distinguishes it from other gram-negative bacteria. Again repeat. Pseudomonas is non-fermenting oxidase positive gram-negative rod. It is non-fermenting oxidase positive gram-negative rod. Non-fermenting oxidase positive gram-negative rod. Non-fermenting oxidase positive gram-negative rod. Okay. Pseudomonas aeruginosa has a polar flagella and pili. This flagella and pili make it mobile. Okay, the most striking bacteriological feature of Pseudomonas aeruginosa is the production of colorful water-soluble pigment. It produces blue pigment that is called pyocyanin. It also produces yellow pigment that is called pyoverdin, or also called fluorescein. It is called fluorescein because it fluoresces under ultraviolet light, and this property of fluorescein can be used in the early detection of skin infections caused by Pseudomonas aeruginosa in burn patients. Okay, pyocyanin and pyoverdin, when combined together, they produce bright green color that diffuses throughout the medium. This striking feature of Pseudomonas also differentiates it from other gram-negative enterobacteria. Okay, the growth of Pseudomonas aeruginosa on a medium has a characteristic metallic sheen, and it emits an intense fruity odor, like the smell of grapes. Okay, the last characteristic feature of Pseudomonas, after which we shall move on to the pathogenesis section, that is, Pseudomonas live in a slime enclosed biofilm. Please see this picture. This is a picture of a lung of a young adult who died of cystic fibrosis. You can see extensive inflammation. You can see in this picture, please focus. You can see extensive inflammation and thick biofilm. This biofilm protects the Pseudomonas from antibodies, from phagocytes. That's why this biofilm allows Pseudomonas aeruginosa to survive well in this biofilm and replicates well in this biofilm and cause chronic infection in patients with cystic fibrosis. Well, this is all about the bacteriology of the Pseudomonas aeruginosa. Let's move on to the pathogenesis of this bacteria, and then we shall move on to the most important section of this illustration video, which is about the clinical features of Pseudomonas aeruginosa. Pathogenesis. Pseudomonas aeruginosa is an opportunistic pathogen. It has a weak invasive ability. That's why healthy individuals don't get infection usually. The healthy individual don't get infection with Pseudomonas aeruginosa usually because the bacteria has weak invasive ability. But it is an opportunistic microbe. Once it is inside a human body, the story changes. What happens in a burn patient? The skin defense system is lost. It takes this opportunity and causes the infection in burn wound. What happens in cystic fibrosis? Patient is immunocompromised. Patient has impaired respiratory tract clearance system. Patient has impaired respiratory tract clearance system, and it takes the opportunity of this impairment and causes the infection. It makes biofilm and produces chronic infection in patient with cystic fibrosis. Similarly, in a patient with immunocompromised status, in immunocompromised patient, it causes infection. It takes the opportunity because the neutrophils are very low in number, and it takes the opportunity and causes the life-threatening severe infections. Once it has taken the opportunity, it produces multiple virulent factors: endotoxin, exotoxin, enzymes, flagella, pili, biofilm, biocyanine, type 3 secretion system. These are all virulent factors due to which or with which it causes infections. Let's elaborate each of them one by one. Endotoxin. Endotoxin causes sepsis and septic shock. Exotoxin. The best known of exotoxin of Pseudomonas aeruginosa is exotoxin A. It causes tissue necrosis. Let me show you how it causes tissue necrosis in this short video clip. Exotoxin A consists of two portions, portion A and portion B. Portion B binds to a receptor present on cell membrane. Portion A is an enzymatic portion that have toxic properties. Once the portion B has binded to a specific receptor on the cell membrane, the whole AB unit internalizes by a process called endocytosis. After the endocytosis, the center of the vesicle becomes acidic. This acidic milia of the vesicle causes the separation of A and B portions. The A portion enters into the cytoplasm of the cell and exerts its toxic effect, while the portion B is removed from the cell by a process called exocytosis. This exotoxin A is now in the cytoplasm. It exerts its toxic effect by stopping translation. The stopping of translation means the stopping of protein synthesis. Let's see how it stops the translation process. This is a ribose and passing through it is messenger RNA. Translation involves the addition of amino acid to the growing peptide chain. and the addition of the amino acid to the growing peptide chain is called elongation. This elongation is facilitated by a factor named elongation factor. There are many elongation factors and one of them is elongation factor 2. And exotoxin A inactivates the elongation factor 2. Once the elongation stops, translation stops. It means the protein synthesis stops. When the protein synthesis stops, cell dies. Means there is necrosis. So exotoxin A causes the necrosis of the cell. And in a short while we shall see a picture of necrotic skin lesion that is caused by Pseudomonas. And this is a manifestation, a clinical manifestation of the action of the exotoxin A. By the way, the mechanism of action of exotoxin A and the mechanism of action of diphtheria toxin is same. But these two toxins, diphtheria toxin and exotoxin A, are different antigenically. Let's stop it here and come on to the next virulent factor. Enzymes. It produces elastases, proteases. These elastases, proteases are histotoxic and these enzymes destroys the body tissue and causes the destruction. This destruction and disruption of the body tissue facilitates the organism to get into the bloodstream, and when it gets into the bloodstream it causes the bacteremia and it causes the sepsis. Type 3 secretion system. Type 3 secretion system, all Pseudomonas aeruginosa don't have this type 3 secretion system. Only some strains have type 3 secretion system. And the strains which have type 3 secretion system are much more virulent than the strains which do not have type 3 secretion system. What exactly type 3 secretion system does? This system transfers the exotoxin from bacteria directly into the adjacent human cell. And the significance of the direct injection of exotoxin lies in the fact that it allows the toxin to avoid neutralizing antibodies. With type 3 secretion system, the exotoxin can avoid neutralizing antibodies. In type 3 secretion system, there are transport pumps in the bacterial cell membrane with which mediates the transport of the toxin. And we can see how these transport pumps operates in this short clip. Before finishing the pathogenesis section with a description of cystic fibrosis, with a description of the pathogenesis of the cystic fibrosis, let's finish quickly the remaining virulent factors. Polar flagella. Polar flagella makes the Pseudomonas mobile and it is critical in causing pulmonary infection. Pili. Pili facilitates the adherence of the Pseudomonas aeruginosa to the epithelial cells. Pyocyanin. Pyocyanin causes the destruction of the cilia and mucosal cells of the respiratory tract.